This video lecture is covering chapter 16, which is the regulation, gene expression, and prokaryotes. And it corresponds with your chapter 16 study guide. And in today's lecture, we're going to cover the concepts of gene regulation and expression and how it responds to environmental conditions. And then we're going to talk in great depth about the lac operon. So how lactose metabolism in E. coli is an inducible system and how that system can also be under a positive control when we introduce glucose into it. So let's begin with prokaryotes and how they regulate their gene expression. This gene expression is in response to environmental conditions. It's been highly studied in E. coli. E. coli has served as a model organism, and because we understand the genome of E. coli, we know where all the genes are, where the promoters are. It's been a highly efficient mechanism to study how these systems have evolved and how transcription of specific genes can be turned on and off depending on the cell's metabolic needs. So those metabolic needs are going to be dictated by the environment. And when the environment changes, then the cellular activity will change to suit that. So with these bacteria that can adapt to their environment by producing certain enzymes, we call those enzymes inducible enzymes. They're not always being made, they're only being made when specific substrates are present. Enzymes, however, that may be continuously produced regardless of what the environmental makeup is, we call those constitutive enzymes. So there's two types of enzymes, inducible and constitutive. Inducible get turned on when we have the substrate. Constitutive are being produced at all times. Sometimes when we have an abundance of the end product in these enzyme systems, then we can have what's called gene repression, in which we repress that system and stop producing the enzyme. Now, regulation of the inducible and repressible types is either under what we call positive control or negative control. Now, negative control is when the expression occurs unless it is shut off by some form of regulatory molecule. So you can think about this as when the molecule is bound to the DNA in a negative control setting, we will not transcribe that gene. In positive control, it's the opposite. Transcription occurs only if a regulator molecule directly stimulates it. So in this case, when something is bound, then typically we will have that production. So negative control, when something's bound, there is no production. Positive control, when something is bound to the DNA, we have the production. Now, in prokaryotes, genes that code for enzymes with regulatory functions are usually organized in clusters, meaning we have genes that are right next to each other. We call these operons. Transcription of an operon is under control of just a single regulatory region. And these regulatory regions are located upstream, that means towards the 5' end of the DNA molecule. And the gene cluster that they control is then going to be under that. In this regulatory region, this is where something will bind, a regulatory protein. We call these regulatory regions cis-acting sites. Now, we're going to be talking about cis and trans acting sites for a couple of lectures. So it's important that you understand what these are. A cis-acting site is a regulatory region that's located on the same strand as the gene cluster. So cis meaning same, right? Trans-acting elements are molecules that bind to cis-acting sites. So trans means opposite. So if you have a cis-acting site, then you will have a trans-acting element that binds onto it. And this can then help regulate, either negatively or positively, the transcription. So let's look at lactose metabolism in E. coli, and we'll kind of look at this as how is it regulated in this inducible system. Now, typically when the lactose is present in the environment, so on a growth medium, if we have lactose, then the concentrations of the enzymes responsible for metabolizing that lactose, we see those concentrations rapidly increase inside the E. coli cell. So we may go from a few molecules of those enzymes to thousands of molecules of those enzymes. And what those enzymes do is they allow the lactose to be broken down into a simple sugar, glucose, that can then be utilized for ATP production. Okay. Therefore, because lactose is the molecule that seems to turn this system on or off, or we want to be careful when I say on and off, because it doesn't actually, lactose doesn't bind to the DNA, but when lactose is present, then we induce the system, so we call lactose the inducer. Okay, so let's look at the lac operon. Again, we have a series of structural genes in our lac operon. There's 3 structural genes, beta-galactidase, permease, and transacetylase. I'll talk about those in a second. We abbreviate these as LAC-Z, LAC-Y, and LAC-A. And you'll see that they are one right after the other. There's nothing in between. Okay. We then have a regulatory region that's located again upstream, so it's towards the 5' end of the DNA molecule, and that regulatory region contains a promoter and an operator, which we'll talk about those in a minute. Okay, and when we transcribe this, we will transcribe all of LAC-Z, LAC-Y, and LAC-A. Okay, we call that constitutive transcription. So let's talk about these different regions. First, the LAC-Z. Okay, this codes for beta-galactidase. And this is an important enzyme in lactose metabolism because what it does is it allows lactose, it breaks the disaccharide lactose into monosaccharides of glucose and galactose. So it breaks this bond. Okay, and we're going to need glucose if we want to utilize it for glycolysis. Okay, LAC-Y and LAC-A. So LAC-Y is permease. This enzyme, it facilitates lactose being brought into the cell, hence the name, permease, to permeate. Okay, and then LAC-A, we don't understand LAC-A as well, but it's hypothesized that transacetylase may be involved in removal of some of the toxic byproducts of lactose digestion from the cell. So when we're done with it, there's some byproducts, and transacetylase is probably being utilized in those processes. And as I said, all 3 genes get transcribed as a single unit. So we get what we call a polycistronic mRNA. And because we're in a prokaryote, we don't have a nucleus in the prokaryote, so our DNA is in the cytoplasm. So as soon as we start transcribing and we start producing our polycistronic mRNA, we can actually start translating as well. So ribosomes can attach to it. And because we get this single mRNA that has 3 parts, what we find is, right, it starts going along, it produces beta-galactidase, it hits a stop codon, it hits a start codon, starts another one. We get permease, hits a stop codon, start codon, starts transcribing another one, and we get transacetylase. So the cistron is the part of the nucleotide sequence that codes for a single gene. This is polycistronic, and 3 gene products are produced. Okay, going back to our operon, we also have even further upstream another gene called the LAC-I. Okay, and LAC-I regulates the transcription of our structural genes. It forms an allosteric repressor protein. When this gets transcribed, we produce a repressor protein, and that repressor protein can interact with the operon itself. So let's look at that. What happens is this allosteric, and by the way, allosteric means that it can when it interacts with another molecule, it's going to change its protein conformational shape. So the three-dimensional shape will change, and that changes its chemical activity. So we produce this repressor protein, and the repressor protein has a binding site. So it has a sequence that will bind to the operator region of our regulatory region. Okay, and it also has a lactose binding site. It's where lactose can bind to. Now, if we don't have lactose present, the repressor protein will bind onto the operator and it'll engage in negative control. So you can kind of think about this as like a wall, right? If we're transcribing, then we're going to have RNA polymerase that's going to attach to this DNA molecule, and it's going to start transcribing. And the promoter region is where we have our TATA box. This is where the RNA polymerase would attach, and then it's going to start transcribing. But if the repressor protein is attached to the operator, it creates this wall, right? So the RNA polymerase can't move past it, and so we will not actually transcribe our lactose operon. We're not going to get Z, Y, or A. Now, if lactose is present, and again, lactose is called the inducer for this very reason, because why? These enzymes that we're producing are needed in lactose metabolism. So if lactose is present, it can bind to the repressor protein, to this binding start, and then we get this allosteric change, right? So when this binds, it changes the 3D folding conformational shape. The protein folds a little bit differently, and now that active site that used to be able to bind at the operator, it can no longer bind there. So what happens is we don't have any binding here. That means that transcription can proceed, and we will get our enzymes. Okay, so again, the repressor protein, when it's bound, transcription is off. So that means it is negative control. Okay, when something's bound to DNA, if we don't produce, negative control. So we can see that the operon model, it has a series of these molecular interactions between the protein, the inducer being lactose, and the DNA, in which we can have this regulation of these structural genes. If we don't have lactose present, then enzymes, they're not needed by E. coli. It doesn't need to produce those enzymes because it would be a waste of energy and to metabolize something that's not present, right? However, if lactose is present, then we are going to induce the activation of those genes so that we can utilize it. So once we've used up all of the lactose and we've metabolized all of that, guess what? There's none that's available to be binding to the repressor, and then transcription is going to get repressed again. So here's this nice little elegant system in which we can turn off these enzymes and turn on these enzymes based off of this repressor protein in the presence or absence of lactose. Now, we may be asking, how do we know all this about the lac operon? How do we know where these repressor protein is, or where the operator site is, or even the structural genes? And a lot of this information came from experiments in which researchers looked at these particular regions and introduced mutations into these regions to see what they would do. So here's an example of a series of experiments that were done that kind of proved how this inducible system was working. So what we see is, here's a genotype. The I stands for the inducer, the O is for the operator, and the Z is for the lac-Z, so the beta-galactidase. And in a normal E. coli, so these would all be wild type, and in the presence of lactose, we don't have much beta-galactidase activity, and beta-galactidase activity can be monitored in this experiment where they just looked at the amount of beta-galactidase present versus not. Okay, so with lactose, we see beta-galactidase activity. Without lactose, we don't see any. Okay, that's the normal system. So then there was a series of mutations that were put in different regions, right? Because we didn't understand what all these regions were. It's not like you could read the DNA and go, oh, that's the operator region. And when they introduced mutations first in the LAC-Z region, what they found is that you never got beta-galactidase activity. So that told them, okay, this region is responsible for this protein or this enzyme, beta-galactidase. When they put a mutation, or when a mutation was present in the region that we call the inducer protein, what we found is that in the presence of lactose, yes, you made beta-galactidase, but when there's also no lactose present, you are also making beta-galactidase. So this told researchers, hey, this region right here appears to be a regulatory region. Something is important. If this isn't functioning, we're always producing this. Likewise, here when we had a mutation in the operator site, they would have beta-galactidase activity both when the lactose was present and when lactose was absent. So again, saying, hey, this region seems to be really important for inducing and turning on and turning off this system. Okay, so then they started looking at, okay, well, maybe does position matter? So they introduced plasmids. Okay, so here we see the E. coli, it has a mutation in the inducer region, so it should function like this, right? Where it's always going to be producing beta-galactidase. But then they introduced through a plasmid, so here's an F' plasmid, an additional copy of a functional inducer gene. And what they found is it restored that normal functioning where in the presence of lactose, we get beta-galactidase, but when lactose is absent, we don't. Okay, so that told them, right, the position of the inducer isn't necessarily all that important, right? This is a protein that's being produced in a combined. As long as that protein gets produced, we're going to have function. Whereas with the operator, they tried the same thing, right? So here's the mutation in the operator, and then they introduced a functional operator on a plasmid. But in this case, they get beta-galactidase activity both when lactose is present and when lactose is absent. So what does that mean? Well, it means that this isn't necessarily producing a protein, right? This is now a regulatory region. We have to have a cis-acting site. This is the cis-acting site. It's on the same strand that we have our functional or structural genes. Whereas this one is just sitting on a plasmid. So even though we have a functional repressor protein and it may bind to this operator, that's not going to stop transcription. So this is how they worked out the difference between the inducer or the operator system. Okay? You can go on and look a little bit more and see, they do things where it's fully functional. This is just kind of to retest B. We have a fully, a wild type E. coli, but then they introduce the F plasmids. In this case, this is where the mutational inducer is, but we still have normal functioning where in the absence of lactose, we don't produce beta-galactidase. And here, likewise, they put a mutational operator in, and again, we get, we still have, or we don't have beta-galactidase activity when lactose is absent. So it doesn't matter if we have mutational forms as long as we have a functional one. Then the cis-acting site for the operator, or somewhere in the system, a inducible one. Okay, and we're gonna just not worry about D. Okay, so again, just going through this, I explained all of this. Your book has some pictures just to show it. So again, this is where we had the mutation in the inducer, and so it wasn't able to attach. And so we would continue to get lactose. It would be constitutive. This is where the operator had the mutation, so we still have it, constitutive. Negative inducible system in the lac operon. Now let's talk about the catabolite activating protein and what the role it plays in positive control in the lac operon. So the catabolite activating protein, or CAP, is a molecule that's utilized during glycolysis. So you may remember CAP and cyclic AMP. Cyclic AMP is really important in the glycolysis process. It's where we're providing phosphate that's used as an energy source in the glycolysis reaction. And so these CAP and CAMP can associate with each other. And in this case, when CAP binds to the lac operon in the regulatory region, then we will have transcription occur. So that means that we have a positive control. So let's look at these two. So again, here's our cyclic, so our catabolic activating protein, and here's our cyclic AMP. And these two will bind to each other. So when we have cyclic AMP, it will bind on to the catabolite activating protein. This changes its conformation into where now it can bind to the promoter region. Okay, it's binding to the promoter, not the operator. And when it binds to the promoter, what this does is it allows for RNA polymerase to actually bind in the promoter region. When we don't have the cyclic AMP and the CAP complex bound, it is very difficult for RNA polymerase to bind in the promoter region. Why? Because the promoter region, it kind of is, if we think about this in the 3D structure, this kind of is wound up on itself. And so the promoter region isn't accessible. We can't get the RNA polymerase in there. Why is this important? Okay, so I just told you that cyclic AMP, it's adenosine monophosphate. Okay, that monophosphate again gets utilized in the creation of ATP, right? So when we have glucose present in a cell, that inhibits the activity of another enzyme called adenylocyclase, which basically converts ATP into cyclic AMP. Why? Because we're going to use this, we're going to go in reverse and make lots of ATP. So when glucose is present, we don't have much cyclic AMP. Instead, we have lots of ATP, right? Why? Because we're utilizing glucose. And then, when without cyclic AMP binding to CAP, CAP can't really bind efficiently to that promoter binding region, which means the RNA polymerase also can't bind very well, which means transcription doesn't stop. So let's think about this for a second. Why is this important for the lac operon? Well, lactose can be broken down by the enzymes, and we generate a single glucose molecule and galactose. And then we can further break down that glucose and generate energy, right? The cell can create ATP. However, if we already have glucose, why do we want to go through the additional step of breaking down lactose just so we can get a glucose molecule? That would be an inefficient use of cellular resources. And so the lac operon has evolved this regulatory region to where when we have glucose present, we're just going to utilize the enzymes needed for glycolysis. We're not going to mess with lactose metabolism. And so therefore, we have this cyclic AMP that can bind here, and this bacteria is going to be much more efficient because it is no longer producing enzymes that it doesn't need. Now, in order for us to have maximal transcription, we have to have the repressor must be bound by lactose. So if we want to get the most transcription process, we have to have lactose present so that it can bind to the repressor so that we don't get that negative control. And we have to have the cap bound to our cyclic AMP at the cap binding site in the promoter. Okay, so we have to not have the negative control, and we have to have the positive control. If we just have cyclic AMP and cap, they're not going to be able to bind to the promoter or to each other. We have to have what we call this cooperative binding. So in the presence of lactose and then with the cyclic AMP, everything can bind together. So we have to have all three of these. And just to reiterate this, we've been looking at everything in a very 2D structure. Let's talk about these regions and try to. I'm going to try to build a mental picture for you of what's actually going on. Right. So here's our regulatory regions. Here's where we have the cap binding site in the promoter. Okay. Here's where we have our operator site. Okay. When the operators are bound, these three sites, then we'll get maximal repression of our operon to where we will not be able to transcribe. And we can see that with the repressor proteins. Okay, so here's the 3D structure. What we're seeing here is here's our DNA, right? And we see that we have this kind of like this loop. This pink molecule right here is the repressor protein. This blue molecule here is cap. And so what you see is when that repressor binds at the operator site, and again, I'm just going to go back. You can see here's an operator site right here. Okay, there's kind of a region right here and right here. But when it binds at this operator site, it creates this loop, right? And this is where the promoter region is. So even if cap and camp were present and they were to bind here, if lactose isn't present, there's no way that RNA polymerase is going to be able to get through this loop and then transcribe the structural genes that would be further down this way. So we call this a repression loop. And this is what is occurring with these bindings. So in the 2D form, you can kind of think about it like we were seeing here, where we have these blockers. So we get blocked. But what's actually happening is that we're creating this repression loop. Okay, this is what I want you guys to know from this chapter, is to really understand the catabolite repression, the inducible enzyme, the negative control, positive control of the lac operon. This chapter goes on to talk about the tryptophan and the arabinose operons. We're just going to focus on lac. So make sure you understand the lac operon, what's going on, what the different regions are, forward and backward. That's what you'll be tested over.